We're pretty excited about the recent data from our phase 1 studies with SON-1010. We've now dosed a total of 72 subjects with SON-1010 with no dose-limiting toxicities or DLTs at any of those doses.
Welcome back, everyone, for another Virtual Investor What This Means segment. Today, we are featuring Sonnet BioTherapeutics, and I'm very pleased to be joined by Raghu Rao. He is Interim Chief Executive Officer, and Dr. Richard Kenney. He's Chief Medical Officer of Sonnet. Welcome, gentlemen.
Janine, thanks for having us.
Absolutely. Well, before we get started, I just want to remind our audience that Sonnet Biotherapeutics is publicly listed on NASDAQ and trades under the ticker SONN. And during today's discussion, the company will be making forward-looking statements, and I encourage everyone to view the company's latest SEC filings on their website at sonnetbio.com for the latest information. So we're going to dive right in here. Dr. Kenney, let's start with an overview of SON 1010. And what exactly is SON 1010, and why does Sonnet believe it's differentiated from other efforts to administer interleukin 12 or IL-12?
Well, thanks, Janine. SON-1010 is a type of cancer immunotherapy that improves the ability of IL-12, which is a naturally occurring protein that's part of the human immune system, to respond to cancer. Even though various mouse models have shown the promise of IL-12 to treat cancer for decades, its use as a cancer therapy in humans has been limited by toxicity. SON-1010 is a fusion protein that consists of unmodified, fully functional recombinant IL-12 attached to a single-chain antibody fragment that binds albumin. We call this our fully human albumin binding technology platform, or FHAB. We've designed this antibody to tightly bind albumin at both normal and at acidic pH, which is typically found in the tumor microenvironment, or the TME. This is really important because we want to deliver and retain the IL-12 in the tumor space to avoid any off-target toxicities. Once inside the TME, the albumin molecules carrying SON-1010 bind to a protein that's called SPARC because tumors use albumin as an energy source for rapid growth, and SPARC tends to keep it there. This mechanism is also used by successful chemotherapies like Abraxane, which is albumin-bound paclitaxel. That drug was designed to bias the delivery and retention of paclitaxel to the tumor based on albumin binding to SPARC. Of course, with SON-1010, we're delivering IL-12 to the tumor instead of chemotherapy. IL-12 binds to receptors on immune cells in the TME, like the NK cells, T cells, and B cells, to activate them, primarily increasing gamma interferon production. This series of events also inhibits regulatory T cells and increases PD-L1 expression, which is the target for the checkpoint inhibitor atezolizumab, or Tecentriq, which we're investigating in a collaboration with Roche in our phase 1b/2a study called SB221. We're also exploring combining SON-1010 with trabectedin, or Yondelis, in certain types of sarcoma, because we think the combination will synergize within tumors to improve the immune response. We believe the mechanism that we created with SON-1010 is differentiated from free IL-12 because the unbound cytokine doesn't have an enhanced PK profile, and it doesn't have preferential uptake or retention within the TME. Some companies are focused on making IL-12 conditionally active, using either a prodrug or masked IL-12 technologies. These are mainly designed to prevent systemic activation of the immune system. But most approaches like this lack SON-1010's preferential uptake and retention in the tumor. Inactivating IL-12 could mitigate some of the side effects, but that doesn't mean you're getting sufficient dose inside the TME. Other companies are developing bispecific proteins, such as the combination of IL-12 and PD-1, but that approach doesn't provide a mechanism for tumor uptake either, and it lacks the flexibility of dosing the drugs separately.
So, Dr. Kenney, you mentioned that safety or side effects have been an issue with IL-12. Can you talk about some of the more meaningful side effects seen with past efforts to dose IL-12 and how SON 1010 is designed to overcome those safety challenges?
Sure, Janine. As we mentioned above, the use of IL-12 as a cancer therapy has been limited because it can have severe toxic side effects. The first phase 1 trial of recombinant IL-12 was actually fine and showed a maximum tolerated dose, or MTD, of 500 nanograms per kilogram. However, when they started with daily dosing in the second trial, they ran into toxicities. The primary adverse effects seen with high dose systemic IL 12 include flu like symptoms, blood abnormalities, liver toxicity, and most notably, a cytokine release syndrome, or CRS. CRS can cause fever, fatigue, nausea, and in severe cases, multi organ failure. Researchers have eventually found ways to dose systemic IL 12 more safely, but they weren't able to dose frequently enough to consistently control tumor growth in humans. Some groups are delivering IL 12 directly into the tumor, which might help avoid some of the systemic toxicities of IL 12. Intratumoral IL 12 does show evidence of local activity, but you need to have a tumor that's accessible for injection. This strategy can miss distant metastases if the immune response is limited to where the IL 12 was delivered. We believe that systemic dosing with an approach that targets all of the tumors, like our SON1010 FHAB, can lead to the highest local IL 12 activity with the least amount of toxicity.
So, Rick, Sonnet is currently conducting phase one studies with SON 1010. And you've mentioned earlier that the company is exploring SON 1010 as a monotherapy, but also in combination with atezolizumab in collaboration with Roche and with trabectedin in sarcoma. Can you provide an update on what the company has seen so far from these studies?
We're pretty excited about the recent data from our phase 1 studies with SON 1010. We've now dosed a total of 72 subjects with SON 1010 with no dose limiting toxicities or DLTs at any of those doses, including 23 healthy volunteers, 24 cancer patients with the monotherapy, 6 sarcoma patients dosed with trabectedin, and 19 patients with advanced tumors or PROC who were dosed in combination with atezolizumab in ovarian cancer. A total of 15 patients have been dosed at the MTD of 1,200 nanograms per kilogram, and the safety profile continues to be similar to that of a dose of IL 12 up to the MTD. Overall, 18 of the 39 evaluable patients to date have had clinical benefit at 4 months, and 2 of the 15 patients dosed at the MTD have had PRs, including one with clear cell sarcoma and one with ovarian cancer. In February, we presented a poster at the AACR IO conference that highlighted the advantages of the FHAB platform, showing long serum half life and concentration in the tumors. We think we're right on target with the therapeutic profile that we envisioned for SON 1010. At the highest dose in the first study, we had an 83% clinical benefit rate at 4 months. This is very encouraging because it shows our drug is active as monotherapy, which is a characteristic that FDA says is important for cancer medications. In addition, our data from the combination with atezolizumab is beginning to show a therapeutic effect at the MTD as well. In the past few weeks, we've released safety updates for SON 1010 monotherapy and from the combinations with trabectedin in sarcoma and with atezolizumab in ovarian cancer. We've not seen unexpected toxicities in either case, and most adverse events have been mild or moderate, suggesting no added side effects from these two combinations. Our safety review committee has reviewed all the existing data from our atezolizumab combination study and approved the progression into the dose expansion phase of that study. We've not seen any DLTs or CRS in that study, and we've only had one related SAE that was a grade 2 pneumonitis, which is a known side effect with atezolizumab. So we're really very encouraged with what we've seen so far with this combination. A third of the patients have achieved clinical benefit in the study, and one of the first 3 ovarian cancer patients at the highest dose actually had a partial response. Overall, we're looking forward to the next steps and feel that IL 12 holds a tremendous promise as an immunotherapeutic agent. That's why you see so many companies with IL 12 development programs. We believe that we've got something special here because we've designed SON 1010 to have extended PK. We have evidence we've successfully targeted the TME, and we're starting to see strong tumor responses at the MTD.
Rick, very helpful. Thanks for breaking that down for us. And definitely an exciting time for the SON 1010 program. So, Raghu, before we end, what are the next steps for the SON 1010 program?
Sure, Janine. We view the current safety and efficacy results we just released as really pivotal milestones that strongly validate our potential of the SON 1010, which is our lead asset. With the MTD now established, both as monotherapy and in combination, we are encouraged by the significant tumor responses observed in both settings. This progress really positions us to confidently advance into the next phase of development, where we plan to generate statistical evidence of the clear benefit of SON 1010 in combination with atezo in ovarian cancer and with trabectedin in soft tissue sarcoma. We are energized by the momentum and very eagerly anticipate sharing results for these studies in the second half of this year.
Well, we'll certainly look to have you back to share those results on our platform. Gentlemen, thank you so much. And this does conclude our Virtual Investor What This Means segment featuring Sonnet BioTherapeutics. I'd like to thank Raghu Rao and Dr. Rick Kenney for joining us today. And I encourage you to visit sonnetbio.com for more information on the company and to follow your social channels to stay current on the latest information. And you can also visit virtualinvestorco.com for our latest segments and upcoming events. Thanks, everyone, and wishing you a great rest of your day.